Right now, an invisible war is being fought all around you. It's on your skin, on the surface of your phone, and deep inside your gut. Trillions of microscopic entities are constantly fighting for survival. For billions of years, bacteria have mastered the art of adapting, defending, and invading. But to us, they aren't just fascinating microbes. In the world of modern medicine, they can be a matter of absolute life and death. When a patient rushes into an emergency room with a raging, unknown infection, doctors don't have days to wait for complex genetic sequencing. They need answers in minutes. And the very first test they run relies on a breakthrough made all the way back in 1884 by a Danish bacteriologist named Hans Christian Gram. Gram discovered a simple chemical staining technique that splits almost the entire bacterial kingdom into two massive, opposing factions, gram-positive and gram-negative. But this isn't just an arbitrary color code. That single difference in color reveals a completely different fundamental design in microscopic engineering. It tells doctors how the bacteria breathe, how they defend themselves, and most importantly, which antibiotics will successfully destroy them, and which ones will fail completely. In this video, we are going to tear down these cellular walls atom by atom. We will explore the brilliant biochemistry of their armor, look step by step at the lab experiment you can do yourself, and uncover why one of these factions holds a terrifying weapon that can trigger fatal shock in the human body. Welcome to the ultimate breakdown of gram-positive versus gram-negative bacteria. To understand why bacteria react differently to a laboratory stain, we have to look at their architecture. Just like human castles required thick stone walls to withstand siege engines, bacteria require a rigid outer structure to withstand immense internal osmotic pressure. Without a strong wall, a bacterium would literally absorb water from its environment and pop like an overinflated balloon. The magical material responsible for this strength is called peptidoglycan, also known as muriin. Think of peptidoglycan as a microscopic molecular chain-link fence. It is composed of alternating sugar chains made of N-acetylglucosamine, or NAG, and N-acetylmuramic acid, or NAM. These long sugar tracks are cross-linked together by short, sturdy chains of amino acids, creating a highly stable, mesh-like fabric wrapped around the entire cell. But how this fabric is laid out is where our two bacterial factions completely diverge. Let's look at faction number 1, the gram-positives. Gram-positive bacteria decided to put all their defensive points into raw thickness. Their cell wall is a monolithic, multi-layered fortress. It features a massive stack of peptidoglycan layers, measuring anywhere from 20 to 80 nanometers thick. This dense mesh accounts for up to 90% of the cell wall's dry weight. Threading through this thick jungle of sugars are unique molecules called teichoic acids and lipoteichoic acids. Think of these as long, reinforcing steel rebar rods driven vertically through concrete. Teichoic acids stick out past the surface, giving the gram-positive bacteria a strong negative charge, helping them bind to surfaces, and acting as a scanning highway for nutrients. Beneath this giant wall lies a single, standard plasma membrane. Simple, heavy, reinforced armor. But now, let's look at the master design of faction number 2, the gram-negatives. Gram-negative bacteria took a completely different evolutionary path. Instead of building a thick wall, they built a sophisticated, multi-layered security system. If you look at a gram-negative cell wall under an electron microscope, you'll notice its peptidoglycan layer is incredibly thin, often just a single layer or two, measuring a measly 2 to 3 nanometers thick. That is a fraction of the gram-positive wall. If that was their only defense, they would perish instantly. But the gram-negative genius lies in what sits above that thin wall, a completely secondary, highly specialized lipid bilayer called the outer membrane. This means the thin peptidoglycan layer is safely sheltered inside a fluid-filled gap called the periplasmic space, sandwiched perfectly between an inner plasma membrane and this tough outer membrane. This outer membrane isn't just a passive skin, it is asymmetrical and heavily armed. The inner leaf of this membrane is made of standard phospholipids, but the outer face is packed with complex, hair-like structures called lipopolysaccharides. So, to summarize the architecture, gram-positive is a single membrane protected by a massive, exposed brick wall. Gram-negative is a thin wire fence hidden securely between two high-tech, heavily guarded electronic gates. This brings us to the famous test that exposes these architectural blueprints in real time, the Gram stain. How exactly does a series of chemical rinses reveal a cell's hidden structure under a basic light microscope? Let's walk through the four-step laboratory protocol to see the fascinating molecular chemistry at play. This brings us to the famous test that exposes these architectural blueprints in real time, the Gram stain. How exactly does a series of chemical rinses reveal a cell's hidden structure under a basic light microscope? Let's walk through the four-step laboratory protocol to see the fascinating molecular chemistry at play. Crystal violet is a basic, positively charged dye. Because both gram-positive and gram-negative cells have negatively charged surfaces, the purple dye molecules easily penetrate the cell walls and flood the cytoplasm of both types of bacteria. At this exact moment, if you looked under a microscope, every single cell on the slide would look identical, a deep, royal purple. Step 2 is the application of a mordant. The slide is rinsed and flooded with a solution of grams iodine. In chemistry, a mordant is a substance that sets or fixes a dye. Inside the cells, the iodine molecules bind directly to the crystal violet molecules, chemically reacting to form a massive, insoluble compound called the CVI complex. This complex is physically much larger than the original dye molecules, meaning it is now trapped inside the mesh of the cell. Both bacteria remain dark purple. Step 3 is the critical fork in the road, decolorization. The scientist tips the slide and pours a rapid rinse of 95% ethanol, or acetone, over the cells for just a few seconds. This is where the physics of their walls changes everything. In gram-positive bacteria, the heavy alcohol acts as a powerful dehydrating agent. It causes that massive, thick peptidoglycan mesh to instantly shrink, compress, and tighten up. Think of it like a fishing net shrinking in the wash, the holes become microscopic. The giant CVI purple dye complexes are permanently locked inside this collapsed fortress. The purple color cannot escape. But look at the gram-negative bacteria. The alcohol acts as a solvent. It instantly dissolves the lipids making up that sleek, delicate outer membrane, punching it full of holes. Because the underlying peptidoglycan wall is so thin and porous, it cannot hold onto the dye. The CVI complex easily washes right out into the sink. In a matter of seconds, the gram-negative bacteria become completely colorless and invisible once again. Step 4 is the counterstain. To see the now invisible gram-negative cells, the slide is flooded with a final red dye called safranine. Because the gram-positive cells are already saturated and tightly packed with dark purple dye, the light pink safranine has no visual effect on them, they remain purple. But the empty, colorless gram-negative cells readily absorb the safranine, glowing a vibrant pink or red. When the scientist blots the slide dry and looks through the lens, the mystery is solved. Purple means gram-positive. Pink means gram-negative. With a simple four-step chemical dance, the invisible architecture of the microworld is laid bare. But this structural difference isn't just an elegant chemistry trick. For a human being infected by these microbes, the composition of these walls dictates exactly how your immune system reacts, and the type of chemical warfare the bacteria can wage against you. Let's look closer at the outer membrane of the gram-negative bacteria. As we mentioned, it is studded with a molecule called lipopolysaccharide, or LPS. LPS is made of three distinct parts: a long outer sugar chain called the O antigen, a core polysaccharide, and a specialized lipid tail buried deep inside the membrane called Lipid A. Lipid A is one of the most dangerous compounds in the microbial world. It is an endotoxin. The word endotoxin means it is an intrinsic, structural part of the bacterial cell itself. While the bacterium is alive and healthy, Lipid A stays tucked away quietly. But when the bacterium dies, reproduces, or is torn apart by your immune system, massive amounts of Lipid A are shed into the human bloodstream. To your body, Lipid A is an absolute red alert. Your immune cells possess specialized receptors called toll-like receptors, specifically TLR4, which are hardwired to detect even a single molecule of Lipid A. When TLR4 detects Lipid A, it triggers a catastrophic immune overreaction known as a cytokine storm. The immune system panics. It orders blood vessels across the entire body to dilate and become highly porous to let immune cells through. But when this happens system-wide, blood pressure drops catastrophically. Oxygen can no longer reach vital organs. This terrifying medical emergency is known as endotoxic shock or septic shock, and it carries a mortality rate of up to 40%. The very structural component that protects gram-negative bacteria becomes a chemical landmine for the human host. Gram-positive bacteria lack an outer membrane, so they do not have endotoxins. Instead, they rely primarily on exotoxins. Exotoxins are toxic proteins that are actively manufactured inside the live bacterial cell and deliberately secreted out into the surrounding tissue like targeted missiles. Don't underestimate them. Gram-positive exotoxins are some of the most lethal poisons known to science. For example, Clostridium botulinum, a gram-positive rod, secretes botulinum toxin. Just 1 single gram of this crystalline toxin, evenly dispersed, could kill over 1 million people by permanently blocking nerve signals and paralyzing muscles. Is recognized as one of the most lethal biological substances known. Even in extremely small amounts, the toxin is capable of causing severe systemic illness if it enters the body through ingestion, a wound, or inhalation. The toxin works as a zinc-dependent endopeptidase that cleaves specific snare proteins, such as SNAP25, within nerve terminals. This process prevents the release of the neurotransmitter acetylcholine at neuromuscular junctions. Because nerve signals are unable to reach the muscles, the condition results in flaccid paralysis, which can lead to critical respiratory failure. Despite its toxicity, the substance has a beneficial side in clinical settings. When utilized in extremely dilute and precisely controlled doses, botulinum toxin, such as Botox, serves as an effective therapeutic and cosmetic treatment. It is used in medicine to manage conditions like severe muscle spasms, chronic migraines, and other neurological disorders by selectively relaxing overactive muscles. Clostridium tetani releases tetanus toxin, causing painful, violent muscle spasms known as lockjaw. The bacterium Clostridium tetani thrives in low-oxygen environments, like deep wounds, where its spores germinate and release a highly potent neurotoxin called tetanospasmin. The toxin binds to peripheral nerve terminals near the wound and travels via retrograde axonal transport to the central nervous system, CNS. In the CNS, the toxin irreversibly blocks the release of inhibitory neurotransmitters, GABA and glycin. Without these inhibitory signals, motor neurons become hyperactive, causing the continuous, uncontrolled, and painful muscle contractions known as lockjaw trismus. Symptoms typically appear within 3 to 21 days, averaging 8 days, after initial infection. The initial symptom is usually masseter muscle spasm, lockjaw, followed by difficulty swallowing. It advances to rigid facial expressions, rhizus sardonicus, and severe, painful, generalized muscle spasms, sometimes causing the body to arch backward, opisthotonous. So, while gram-negatives carry built-in structural landmines that devastate the body when they die, gram-positives act as active manufacturing plants, pumping out highly targeted, deadly chemical weapons while they live. This brings us to the ultimate question facing modern medicine: how do we kill them? When an infection takes hold, we deploy antibiotics. But an antibiotic that wipes out a gram-positive infection might be completely useless against a gram-negative target, purely because of the physical barriers we have just discussed. Let's look at the classic miracle drug, penicillin. Penicillin belongs to a class of drugs called beta-lactams. Its specific job is to act as a molecular wrench thrown into the gears of bacterial construction. It binds to the enzymes responsible for cross-linking those NAM and NAG sugar chains in the peptidoglycan wall. In a growing gram-positive bacterium, penicillin easily passes through the porous outer layers and shuts down wall production. Without those amino acid crosslinks, the massive wall loses its structural integrity. The intense internal pressure of the cell takes over, water rushes in, and the gram-positive cell literally bursts open and dies. But what happens when you throw standard penicillin at a gram-negative bacterium like Escherichia coli? Absolutely nothing. The penicillin molecules are completely blocked by that hydrophobic, tightly packed outer membrane. The antibiotic can't even get close to the thin peptidoglycan layer hidden underneath. The outer membrane acts as an incredible chemical shield against many large, hydrophobic drugs. So how does a gram-negative cell eat, breathe, or absorb nutrients if its outer membrane is an impenetrable wall? The answer lies in specialized protein tunnels called porins. Porins act as highly selective cellular bouncers. They form narrow, water-filled channels that allow small, hydrophilic molecules like sugars and amino acids to pass through into the periplasm while keeping dangerous, bulky chemicals out. To fight gram-negative bacteria, scientists had to design modified, smaller antibiotics, like ampicillin or piperacillin, that are specifically engineered to sneak through these tiny porin channels. This structural divide creates two distinct rogues galleries in clinical medicine. On the gram-positive side, we face pathogens like Staphylococcus aureus, the cause of severe skin infections, pneumonia, and the terrifying drug-resistant superbug MRSA. Streptococcus pneumoniae, a leading cause of bacterial meningitis and ear infections. Listeria monocytogenes, a dangerous foodborne pathogen that can survive even inside human refrigerators. On the gram-negative side, we face monsters like Escherichia coli, notorious for severe urinary tract infections and dangerous food poisoning. Pseudomonas aeruginosa, a highly opportunistic nightmare that colonizes hospital equipment and infects the lungs of cystic fibrosis patients. Neisseria meningitidis, the rapid, life-threatening cause of epidemic meningococcal disease. Today, the outer membrane of gram-negative bacteria has made them a primary threat in the global antibiotic resistance crisis. Many gram-negative strains are actively mutating their porin channels, making them smaller or modifying their charges so that our latest antibiotics can no longer fit through the doors. Combined with internal pumping systems called efflux pumps, they can actively grab antibiotics that enter the cell and spit them right back out before they can do any damage. When we look at the microscopic world, it is easy to view bacteria simply as germs to be erased. But when you look beneath the microscope, you realize you are looking at some of the most elegantly engineered structures on Earth. Whether it is the brute force, heavy concrete armor of the gram-positive wall, or the hyper-advanced, double-membrane security system of the gram-negative cell, these organisms have spent billions of years perfecting their survival mechanics. Understanding these minute structural differences is what allowed humanity to break the cycle of historic plagues, develop life-saving antibiotics, and keep treating critical infections every single day in hospitals around the globe. If you found this deep dive into microbiology fascinating, make sure to hit that like button, subscribe to the channel, and turn on notifications so you never miss an episode. Now, let me know down in the comments, if you were a bacterium trying to survive a harsh environment, which design would you choose? The thick armor of the gram-positives or the tactical adaptability of the gram-negatives? Thank you so much for watching, and we'll see you in the next video.